So now let's start the chapter 8. Cell, the unit of life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it makes an organism living, or what is that an inanimate thing does not have which a living thing has? The answer to this is the presence of basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others like us composed of many cells are called multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Schilden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodore Schwann in 1839, उसके एक साल बाद, स्वान का मीन्स एक एनिमल होता है, तो इससे रिमेंबर कर सकते हैं अपन, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissue, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and product of cells. Schilden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow in 1855 first examined that cells divided and new cells are formed from pre-existing cells. Omnis cellula is cellula. He modified the hypothesis of Schilden and Schwann to give the cell theory a final shape. Cell theory as understood today is all living organisms are composed of cells and product of cells. Second point, all cell arises from pre-existing cells. An overview of cell. You have earlier observed cells in an onion peel and or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cell of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosome, which in turn contain the genetic material, DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are prokaryote. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of the cellular activities in both plant and animal cells. यहाँ पे सिंगल कर सकता है, इन ओनली एनिमल कर सकता है या फिर प्लांट कर सकता है, जो करेक्ट है वो इन बोथ प्लांट एंड एनिमल सेल्स. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structure called organelles like the endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, that is chloroplast in plant and mitochondria and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelles called centrosome, which help in cell division. Non-membrane bound organelle कौन है? Centrosome. Animal cell contain another non-membrane bound organelle called centrosome, which help in cell division. Cell differs greatly in shape, size, and activities. For example, mycoplasmas, the smallest cells, are only 0.3 micrometer in length, while bacteria cells could be 3 to 5 micrometer. Understood? Could be 3 to 5 micrometer बैक्टीरिया, ठीक है. अब माइकोप्लाज्मा का 0.3 माइक्रोमीटर इन लेंथ, याद रहेगा. चलो ठीक है, आगे बढ़ते. ये डायग्राम पढ़ देता हूँ मैं, ठीक है. रेड ब्लड सेल्स होते हैं राउंड एंड बाइकॉनकेव. वाइट ब्लड सेल्स का शेप होता है अमोइबीड. कॉलमनार एपिथेलियल सेल्स होते हैं लॉन्ग एंड नैरो. और नर्व सेल होते हैं ब्रांच्ड एंड लॉन्ग. ट्रेकियड होती है एलोंगेटेड. मिसोफाइल सेल्स होते हैं राउंड एंड ओवल. The largest single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Human RBC is about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Understood? Next is prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, that is pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shape of the bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, spiralum, spiral. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and function. All prokaryotes have a cell wall. Important line है भाइयों. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasm. Important line है. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genomic DNA. This smaller DNA are called plasmids. The plasmids DNA confers certain unique phenotypic character to such bacteria. One such character is resistant to the antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryote cells except for ribosomes. जैसे eukaryotes में होते हैं वैसे prokaryotes में नहीं होते हैं organelles, बस ribosome को छोड़. ठीक है. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristics of the prokaryotes. They are essentially infoldings of cell membrane. Now, typical bacteria are 1 to 2 micrometer, 1 or 2, ठीक है, एंड PPLO की साइज है अबाउट 0.1 माइक्रोमीटर, वायरसेस की 0.02 से लेकर 0.2 माइक्रोमीटर तक, और टिपिकल यूकैरियोटिक सेल की साइज है 10 से 20 माइक्रोमीटर तक, ठीक है। टिपिकल यूकैरियोटिक 10 से 20 माइक्रोमीटर, वायरसेस की 0.02 से लेकर 0.2 तक, PPLO की अबाउट 0.1, टिपिकल बैक्टीरिया की 1 से 2 माइक्रोमीटर। नेक्स्ट इज सेल एनवेलप एंड इट्स मॉडिफिकेशन। Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of tightly bound 3-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelopes perform distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram, which those that take up the Gram stain are Gram positive and the other that do not are called Gram negative bacteria. Glycocalyx differ in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in other it may be thick and tough called the capsule. The cell wall determines the shape of the cell. Cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing, फटने से और collapse होने से बचाता। The plasma membrane is selectively permeable in nature and interact with the outside world. Plasma membrane is selectively permeable. This membrane is similar structurally to that of the eukaryotic. This membrane is similar structurally to that of the eukaryotes. कौन सी? Plasma membrane, ठीक है। A special membranous structure is the mesosome, which is formed by the extension of the plasma membrane into the cell. This extension are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to the daughter cells. They also help in respiration, secretion process to increase the surface area of the plasma membrane and the enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extension into the cytoplasm called chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called a flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of 3 parts: filament, hook, and basal body. Bacteria show a range in the number and arrangement of flagella. Bacterium flagella is composed of 3 parts, that is filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. सबसे longest portion होता है filament and extend from the cell surface to the outside. Beside flagella, pili and fimbriae are also surface structure of the bacteria but do not play a role in the motility. Do not की जगह change करके play कर देगा और do not काट देगा, but correct है do not play, ठीक है। assertion region सब बना रहा हूँ। The pili are elongated tubular structures made of special protein. The fimbriae are small bristle-like fibers sprouting out of the cells. In some bacteria, they are known to help attach the bacteria to rocks in streams and also the host tissues. In some bacteria, they are known to help attach the bacteria to rocks to rocks in streams and also the host tissues. Next is ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S. Consequence subunits 50S and 30S units, which when present together form 70S prokaryotic ribosomes. इसे याद कर सकते हैं, 50 और 30 मिला के 80 बना रहा है तो उनकी टुगेदर यूनिट होगी 10 कम यानी 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosome of a polysome translate the mRNA into protein. The ribosome of polysome translate the mRNA into protein. It's an important line. आगे inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. Reserve material inclusion body में store होता है. These are not bound by any membrane system and live free in the cytoplasm. Example: phosphate granules, synophysian granules and glycogen granules. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria. Next is eukaryotic cell. The eukaryotic includes all the protist, plant, animal and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possesses an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. नींद तो नहीं आ रही है ना? हाँ, उठ जाना, ठीक है? नींद आ रही हो तो. Their genetic material is organized into chromosome. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. Their genetic material is organized into chromosome. All eukaryotic cells are not identical. It's a true assertion region में आ सकता है, ठीक है, या फिर किसी में भी आ सकता है. All eukaryotic cells are not identical. Plant and animal cells are different as the former possesses cell walls, plastids and a large central vacuole which are absent in animal cell. Animal cell में क्या क्या absent होती है? Cell wall, plastid, large central vacuole. On the other hand, animal cells have centrioles which are absent in almost all plant cells. All जहाँ आ जाता है important हो जाता है. Let us now look at individual cell organelles to understand their structure and functions. So let's proceed further. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950. Advent of electron microscope. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells (RBCs), enabled the scientists to deduce the possible structure of the plasma membrane. This study showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outside and the hydrophobic tail towards the inner part. हाइड्रोफोबिक मतलब डर रही है इसलिए इनर पार्ट में, फोबिक का मीन्स डर, ठीक है, ऐसे याद रख सकते हो. दिस एंश्योर्स दैट द नॉन पोलर टेल ऑफ द सैचुरेटेड हाइड्रोकार्बन इज प्रोटेक्टेड फ्रॉम द एक्वस एनवायरनमेंट. हर सबसे बचा के रख रखा है. इन एडिशन टू द फॉस्फोलिपिड मेम्ब्रेन ऑल्सो कंटेन कोलेस्ट्रॉल. कोलेस्ट्रॉल भी होता है भैया. Later, biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipid. 52% protein, 40% lipid. Understood? Depending on the age of extraction, membrane proteins can be classified as integral and peripheral. Peripheral protein lie on the surface of membrane, while the integral protein are partially or totally buried in the membrane. Partially or totally. An improved model of the structure of the cell membrane was proposed by Singer and Nicholson. किसने प्रपोज किया है? Singer and Nicholson in 1972. Widely accepted as fluid mosaic model. Widely accepted as fluid mosaic model in 1972. According to this, the quasi-fluid nature of the lipid enables lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as fluidity, known as fluidity. Understood? So now let's proceed further. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. Next is, one of the most important functions of the plasma membrane is the transport of the molecules across it. And function is most important function of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solid molecules may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from high concentration to lower concentration. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to higher concentration. Such transport is an energy dependent process in which ATP is utilized. ATP is utilized and is called active transport. Example, sodium potassium pump. Next is cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protect the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans, mannans, and mineral like calcium carbonate, while in other plant it consists of cellulose, hemicellulose, pectins and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth which gradually diminishes as the cell matures and the secondary wall is formed on the inner towards membrane side of the cell. In other plant, cell wall consists of cellulose, hemicellulose, pectins and protein. डबल पड़ती है भेजे में घुसा. The middle lamella is a layer mainly of calcium pectate. ठीक है, कंफ्यूज कर देगा और कैल्शियम कार्बोनेट देगा, बट करेक्ट है calcium pectate, which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata, which connect the cytoplasm of neighboring cell. Next is endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as endomembrane system because their functions are coordinated. Assertion reason बन सकता है यहाँ से. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosome and vacuole. Endomembrane system include GUL means Golgi complex, endoplasmic reticulum and lysosome and vacuole. Since the function of the mitochondria, chloroplast and peroxisome are not coordinated with the above component, they are not considered as a part of endomembrane system. Important line है, ठीक है. The endoplasmic reticulum, that is ER, electron microscopic studies of eukaryotic cells reveals the presence of a network or reticulum of tiny tubular structure scattered in the cytoplasm that is called endoplasmic reticulum. Hence, the endoplasmic reticulum divides the intracellular spaces into two distinct compartments, that is luminal inside endoplasmic reticulum and extraluminal, that is cytoplasm compartments. The endoplasmic reticulum often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum. In the absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum. In the absence of ribosome, they appear smooth. कैसी appear होती है? Smooth. ठीक है, or called smooth endoplasmic reticulum. Next is rough endoplasmic reticulum is frequently observed in the cells actively involved in protein synthesis and secretion. किसमें? Protein synthesis and secretion. ठीक है, उल्टे मत जाना. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in the smooth endoplasmic reticulum. In animal cell, lipid-like steroidal hormones are synthesized in ACR. ठीक है. Next is Golgi apparatus. Camillo Golgi in 1898 first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat, disc-shaped sacs or cisternae of 0.5 micrometer to 1.0 micrometer diameter. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with the distinct convex that is VC और the forming face. Convex जो होता है forming face होता है, सर याद करो. CVC F मतलब convex cis face होता है, forming face होता है, और and concave trans और the maturing face, जिसको याद रख सकते हैं CCTM means concave trans means maturing face. The cis and trans phase of the organelle The cis and trans phase of the organelle are entirely different but interconnected. Are entirely different but interconnected. ठीक है. Next is the Golgi apparatus principally perform the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Material to be packed in the form of vesicle from the endoplasmic reticulum fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. Means cis means forming phase के साथ फ्यूज होता है और किस तरफ मुड़ता है, मैच्योरिंग फेज मतलब कॉनकेव तरफ. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosome on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its transphase. Golgi apparatus is the important site of formation of glycoprotein and glycolipids. Next is lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolysis, lipases, proteases, carbohydrates, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acid. Next is vacuoles. The vacuole is the membrane-bound species found in the cytoplasm. It contains water-safe, excretory product and other material not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuole can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other material against concentration gradients in the vacuole into the vacuole. Hence, their concentration is significantly higher in the vacuole than the cytoplasm. Their concentration is significantly higher in the vacuole than in the cytoplasm. In plant, the tonoplast facilitates the transport of number of ions and other material against concentration gradient into the vacuole. Hence, concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particle. Next is mitochondria. Mitochondria is known as the powerhouse of cell. बचपन से अभी तक का फेमस डायलॉग है ना. Mitochondria, that is singular mitochondrion, unless especially stained, are not easily visible under the microscope. The number of mitochondria per cells is variable depending on the physiological activity of the cells, in terms of shape and size also. Considerable degree of variability is observed. Typically, it is a sausage-shaped or cylindrical having a diameter of 0.2 to 1 micrometer, average 0.5 micrometer, and length that is 1 to 4.1 micrometer. And length is 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is, the outer compartment and the inner compartment. The inner compartment is filled with a dense, homogeneous substance called the matrix. The inner compartment is filled with a dense, homogeneous substance called the matrix. Inner compartment is filled with dense, homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms the number of enfoldings called the cristae, singular called crista, towards the matrix. The crista increases the surface area. Crista increases the surface area. Spelling बताओ, CRISTAE, increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial functions. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. The matrix also possesses single circular DNA molecules, a few RNA molecules, ribosomes, that is 70S, and the components required for the synthesis of protein. The mitochondria divide by fission. The matrix also possesses single circular DNA molecules, a few RNA molecules, ribosomes, कौन सी कैटेगरी का 70S का, ठीक है. Next is plastids. Plastids are found in all plant cells and in euglenoids. They are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting a specific color to the plant. Imparting a specific color to the plant. Imparting a specific color to the plant. Based on the type of pigment, plastid can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigment, which are responsible for trapping light energy, essential for photosynthesis. Are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigment like carotene, xanthophylls, and other are present. This gives the part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastid of varied shapes and sizes with stored nutrients. Amyloplasts store carbohydrate, that is starch. Amyloplasts store carbohydrate, that is starch. Example, potato. Elioplast store oils and fats, whereas the alleloplast stores protein. Alleloplast stores protein. Majority of the chloroplast of the green plant are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length, that is 5 to 10 micrometer, and width, that is 2 to 4 micrometer. Their number varies from 1 per cells of the Chlamydomonas, a green algae, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stakes like the piles of coins called grana, singular granum, or the integral thylakoids. In addition, there are flat membranous tubules called the stromal lamellae connecting the thylakoids of the different grana. The membrane of the thylakoid encloses a space called a lumen. The stroma of the chloroplast contain enzymes required for the synthesis of carbohydrate and protein. Stroma of the chloroplast contain enzyme required for synthesis of carbohydrate and protein. It also contains small double-stranded circular DNA molecules and ribosome. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigment are present in the thylakoids. The ribosome of the chloroplast are smaller, that is 70S, than the cytoplasmic ribosome, that is 80S. Cytoplasmic ribosome are 80S. Next is ribosomes. Ribosomes are the granular structure first observed under the electron microscope as dense particle by George Palade in 1953. ऑब्जर्व अंडर था कौन से माइक्रोस्कोप में? इलेक्ट्रॉन की सिंपल टेरेस्ट्रियल, करेक्ट है, इलेक्ट्रॉन माइक्रोस्कोप. They are composed of ribonucleic acid, that is RNA, and proteins, and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. अभी पहले भी आपको बताया था. Each ribosome has two subunits, a larger and a smaller subunit. The two subunits of 80S ribosome are 60S and 40S, while that of 70S ribosome are 50S and 30S. Here S, that is Sievertberg's unit, stands for the sedimentation coefficient. It is the indirect measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. दोनों को मिला के जो आएगा ना उसका कम वाला, ठीक है. जैसे 60 और 40 जो यूनिट है वो 100 बना रही है तो उसकी जो लार्जर यूनिट है 80S होगी, ठीक है. और 50 और 30 प्लस करके 80 होता है तो उसकी जो लार्जर यूनिट है 10 कम 70S होगी, ठीक है. मीन्स सिंपल वे में 70S के 50S एंड 30S है और 80S राइबोसोम की जो यूनिट है 60 एंड 40 है. क्लियर. नाउ नेक्स्ट इज साइटोस्केलेटन. An elaborate network filamentous proteinous structure consisting of microtubules, microfilaments and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. First is mechanical support, motility and maintenance of the shape of the cell. Okay. Next is cilia and flagella. Cilia singular, cilium, and flagella singular, flagellum are hair-like outgrowths of the cell membrane. Cilia are small structure which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possesses flagella, but these are structurally different from that of eukaryotic flagella. The prokaryotic bacteria also possesses flagella, but these are structurally different from that of eukaryotic flagella. Understood? Okay. The electron microscopic study of the cilium or the flagellum shows that they are covered with plasma membrane. Their core, called the axonema, possesses a number of microtubules running parallel to the long axis. The axonema usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. धीरे धीरे इसलिए पढ़ रहा हूँ मैं ताकि आपके माइंड सेट होता जाए. 1 hour lines में speed में. The axonema usually has a 9 doublets of radially arranged peripheral microtubules in a pair of centrally located microtubules. Such an arrangement of axonemal microtubules is referred to as the 9+2 array. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. The peripheral doublets are also interconnected by linkers. Both the cilium and the flagellum emerge from the centriole-like structure called the basal body. Both cilium and flagellum emerge from centriole-like structure called the basal body. Next is centrosome and centrioles. Centrosome is an organelle usually containing 2 cylindrical structures called centrioles. Centrosome is an organelle usually containing 2 cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar material. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplet are also linked. The central part of the proximal rind of the centriole is also proteinous and called the hub. The central part of the proximal rind of the centriole is also proteinous and is called hub, which is connected with the tubules of the peripheral triplets by radial spokes made of protein. Of the peripheral triplet by radial spokes made of protein. The centrioles form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division in animals. The centrioles form the basal body of cilia or flagella and spindle fiber that give rise to spindle apparatus during cell division in animals. So the next is nucleus. Nucleus as a cell organelle was first described by Robert Brown as early in 1831. Later, the material of the nucleus is stained by the basic dyes was given the name chromatin by Fleming. The interface nucleus of a cell, when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of 2 parallel membranes with space between 10 to 50 nanometer, 10 to 50, not 15, 10 to 50, called the perinuclear space. 10 और 15 में कंफ्यूज मत हो जाना, 10 और 50 हो नैनोमीटर. ठीक है, अगर नींद से उठना हो तो बुक में देख सकते हो एनसीईआरटी में. ठीक है, पेज नंबर बता देता हूँ, 100. ठीक है. Called the perinuclear space forms a barrier between the materials present inside the nucleus and that of the cytoplasm, which consists of 2 parallel membranes with a space between 10 to 50 nanometer called perinuclear space forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bear ribosomes on it. At a number of places, the nuclear envelopes is interrupted by minor pores, which are formed by the fusion of its two membranes. These nuclear pores are the passage through which movement of RNA and protein molecule takes place in both directions between nucleus and the cytoplasm. At a number of places, the nuclear envelopes interrupted by the minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passage through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus, that is, erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? क्या consider करोगे? So next is the nuclear matrix or the nucleoplasm contain nucleolus and chromatin. Nuclear matrix or the nucleoplasm contain nucleolus and chromatin. The nucleoli are a spherical structure present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interface nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. You may recall that the interface nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in places of the nucleus. But during different stages of the cell division, cells show structured chromosome in place of the nucleus. Chromatin contain DNA. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins and also RNA. इधर उधर कहाँ सोच रहे हो? आ जाओ सुनो. Chromatin contain DNA and some basic protein called histones, some non-histone proteins and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46, that is 23 pairs chromosome. You will study the details of DNA packaging in the form of chromosome in class 12. But class 12 से क्या करें? मैं तो खड़ा हूँ आज भी वहीं. Let's move forward. Every chromosome visible only in the dividing cells essentially as a primary constriction or the centromere. Primary constriction or centromere, on the sides of which disc-shaped structure called kinetochores are present. Centromere holds two chromatids of a chromosome. Based on the position of the centromere, the chromosome can be classified into 4 types. The metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric chromosome has terminal centromere. Acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm. And in submetacentric chromosome is centromere slightly away from the middle of the chromosome, resulting in one shorter and one longer arm. So next proceed. Sometimes a few chromosomes have non-staining secondary constriction at a constant location. This gives the appearance of small fragments called the satellite. Sometimes few chromosomes have non-staining secondary constriction at a constant location. This gives the appearance of a small fragment called the satellite. Next is microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. So next we are going to read the summary in a speedy way. All organisms are made of cells or aggregate of cells. Cells vary in their shape, size and activities function based on the presence or absence of membrane-bound nucleus and other organelles. Cells and hence organisms can be named as eukaryotic and prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus and cytoplasm. Plant cells have a cell wall outside the cell membrane. Typical eukaryotic cell consists of cell membrane, nucleus and cytoplasm. Plant cells have a cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates transport of several molecules. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome and vacuoles. All the cell organelles perform different but specific functions. Centrosome and centriole form the basal body of cilia and flagella that facilitate locomotion. In animal cells, centrioles also form spindle apparatus during cell division. Nucleus contains nucleoli and chromatin network. Nucleus contains nucleoli and chromatin network. It not only controls the activities of organelles but also play a major role in heredity. Endoplasmic reticulum contains tubules or cisternae, cisternae. They are of two types, rough and smooth. Endoplasmic reticulum help in the transport of substance, synthesis of protein, lipoprotein and glycogen. The Golgi body is a membranous organelle composed of flattened sacs. SACSO. The secretion of cells are packed in them and transported from the cell. Lysosomes are single membrane bound structure containing enzymes for digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasm or are associated with the endoplasmic reticulum. Mitochondria help in oxidative phosphorylation and generation of adenosine triphosphate, that is ATP. They are bound by double membrane. The outer membrane is smooth and the inner one falls into several crystals. Plastid are pigment containing organelle found in plant cells only important in plants. In plant cells, chloroplasts are responsible for trapping light energy essential for photosynthesis. The grana in the plastid is the site of light reaction and the stroma of dark reaction. The green colored plastid are chloroplasts which contain chlorophyll, while the other colored plastid are chromoplasts which may contain pigment like carotene and xanthophyll. Chromoplasts contain pigment like carotene and xanthophyll. The nucleus is enclosed by nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, cell is a structural and functional unit of life. The nucleus is enclosed by a nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, cell is the structural and functional unit of life. With this, we complete the chapter. Thank you for hearing.